Why do proteins need a molecular chaperones? A newly synthesized protein is useless until it falls into the correct three-dimensional structure. Most people think that gene expression ends once a protein is translated, but translation produces only a linear chain of amino acids. Before the protein can be functional or participate in cell signaling, it must take its precise three-dimensional shape. However, protein folding is not a spontaneous or a risk-free process. Inside the cell, thousands of macromolecules compete for space, creating a highly crowded environment where newly synthesized proteins can misfold or stick together, forming toxic aggregates. That threatens the normal cellular function. Here is where molecular chaperones become essential. They don't determine the final structure of the protein. This information is already encoded in its own amino acid sequence. Instead, they guide the folding process, prevent inappropriate interactions, stabilize folding intermediates, and maintain protein homeostasis or proteostasis. Different chaperone families perform specialized tasks. HPS 70 assists newly synthesized proteins during folding. HPS 90 promotes the maturation of signaling proteins and protein kinase. HPS 60 is known as a chaperone, provides an isolated chamber where proteins can fold safely, while small heat shock proteins temporarily stabilize unfolded proteins during cellular stress until they can be properly refolded. Although these mechanisms are highly conserved across life, their biological roles reflect the needs of each organism. In plants, molecular chaperones are indispensable for surviving heat, drought, salinity, and oxidative stress. They protect photosynthetic and respiratory proteins within chloroplast or mitochondria, while co-chaperones such as BAG and HOB integrate stress responses with hormone signaling, growth, and program cell death. In animals, the same molecular machinery protects neurons from the accumulation of misfolded proteins associated with aging and neurodegenerative diseases such as Alzheimer's. When the chaperone network becomes overwhelmed, proteostasis collapses, leading to protein aggregation and cellular dysfunction. In summary, from protein folding to stress tolerance and disease prevention, molecular chaperones are far more than folding assistants. They are essential guardians of proteome integrity, ensuring that genetic information is transformed into functional biologically active proteins.